The digestive pathway. We were still in the mouth talking about our different accessory organs that are found in the mouth. We'd started our process in big picture mode, saying, oh, okay, we need to know the different processes that are involved in the digestive system and where they occur and what they mean. We needed to understand that the alimentary canal has that uniform, to a certain extent, structure to it with our 4 different layers, our mucosa and submucosa and muscularis and serosa. We talked a bit about the control and coordination of the digestive system. So like usual, we're going to find that there is going to be extrinsic control, so that means neural and hormonal. We'll talk more about hormones along the way. In terms of neural, right, this system is one where we don't have to think about things. We don't have to consciously go, oh, I got to swallow, keep going, keep pushing, pooh, made it, right? It just kind of takes over once you do that initial voluntary swallowing. We have peristalsis that moves it through and different types of mixing movements, etc. So autonomic nervous system is going to be the main one in control for neural extrinsic control. So that is mainly parasympathetic, right? Parasympathetic, what's the phrase that goes with it?
Rest and digest.
Rest and digest. So, whereas in some of our other systems, like respiratory system and cardiovascular system that we've seen, parasympathetic would tend to slow it down, and sympathetic would speed it up, like heart rate, etc. In this case, parasympathetic stimulation to the different organs produces secretion, increased smooth muscle activity, etc. And sympathetic stimulation, right? Fight or flight, that slows down the digestive system. If you're in stress mode, if you're running from the bear or something like that, you can digest food later. You can allocate energy to digesting food if you survive the bear attack. Then you can switch over to parasympathetic and get those things going again, right? So you all have experience with stress, various types, physical, emotional, whatever, that can affect your digestive system because that sympathetic is kind of stronger output as opposed to parasympathetic, right? So we did talk about neural control, hormonal control. We'll see that coming up a bit more. And then there's that local intrinsic control, that enteric nervous system, where we said that different sections of this disassembly line can communicate with each other to coordinate the activity along the way, okay? Okay. So those are big picture items. Now we're moving our way through like a particle of food. We had gotten to the mouth. We had talked about the tongue. We had talked about, we didn't talk about the teeth yet. We're coming up to that. And so we were just visiting with salivary glands as well. So we know a decent amount about salivary glands just from our own experience, that the saliva that is produced there helps to clean the mouth, helps to moisten and dissolve the food chemicals. We did talk about that, how we need that saliva to mix with the food so it can kind of drip down to the taste buds, the chemoreceptors that are on the sides of some of those different types of papillae. And then also, as we're mixing it around, we're doing some of that physical digestion, we're creating kind of this mass or ball. That is what's called a bolus. So we're creating some sort of softened bolus that we have physically digestion, and once we have processed it to the level that we think is appropriate, we voluntarily push it back to the pharynx. Pharynx and the esophagus voluntarily then send it down towards the stomach. Okay. In addition to aiding in mechanical digestion and chemosensation and things like that, saliva also has one of our different digestive enzymes in it. So this is salivary amylase. Amylase helps to digest amylose, which is starch, which is a complex carbohydrate. Okay? So we're starting to see just the beginnings of chemical digestion here in the mouth. Little bit of our lipid digestion, because we have a lingual lipase. Little bit of now carbohydrate digestion, because we have a salivary amylase. So it's just going to start that process, but as we are becoming more and more aware, it definitely takes time. Like those exercises, those simulations that we're doing in lab, the incubation period is important to allow these enzymes to do their job. So we're just starting the process here, but we're going to continue it as we move through as well. Okay? One of the things that I wanted to mention as well that I forgot to mention at the beginning is looking at the roles of saliva and salivary glands. I did go through the exam objectives for exam number 3. So I kind of went through and I kind of cleaned it up and in some cases shortened it up and stuff like that. Made sure that the ones that are bold are still going to align with your short answer questions, etc. So I updated all the links that I could find in Canvas. So if you go to your exam objectives now, if you haven't yet, then those should be the new copy. If you did before, then do kind of refresh the page and go review those exam objectives. So they're quite a bit more concise now for preparing for exam 3. Okay, so our functions for saliva and the salivary glands, I remember was kind of in there as well. All right, so here we can see some of our anatomy. We've already done a bit about this in lab, but we know we have 3 major salivary glands, parotid ones, which are the largest ones, which again have their excretory duct that dumps near our like our second premolar or so. It's back towards the back of the mouth, and then we have the ducts coming from our other glands, our submandibular and our sublingual, that are going to be found underneath the tongue instead. But they're all going to be secreting. We know that they are made up of a combination of mucus acini and serous acini, and in fact, when we stimulate them, they can be stimulated by both sympathetic and parasympathetic stimulation, like we said. And in fact, it changes what type of secretion you get. So the mucus acini tends to be a thicker secretion, right? Mucus, as you think about it, is a little bit thicker. Serous acini is more watery. So if we are preparing for a meal and you think about your mouth watering or something like that, then parasympathetic stimulation is going to be kind of poking at those different salivary glands, and you produce a more watery secretion that can mix with the food better, that has more of the digestive enzymes in it, etc. If we're just maintaining the inside of the mouth and we're helping to retard bacterial growth and that type of thing, then it's a little bit maybe more of the mucus acini instead. And so that's a little bit more viscous, a little bit less watery, and so that's kind of a balance of sympathetic and parasympathetic stimulation to those glands. But those are our salivary glands that are also then found in the mouth and are helping to create this environment where we can start digesting the food, right? Mostly water, slightly acidic. It does have some electrolytes or salts in it. It does have that salivary amylase in it. And then there are a few, remember, this is part of the first line of defense. Lysozymes, defenses, IgA, that's immunoglobulin. That's an antibody that's secreted there as well. So there's a lot of dirty things that can go in your mouth, especially when you're a toddler, etc. And so we need to be protecting ourselves there. So we do have a variety of different types of protein secretions in there to help protect us. And then oddly enough, there are some metabolic waste products in there. So in sweat and different types of exocrine secretions, we do get some metabolic waste products coming from those cells. Okay, so then we go to the teeth. With the teeth, a lot of the anatomy of the teeth we've gone over in lab, so we won't spend a lot of time with that, right? The different layers, the enamel, the dentin, the cementum, things like that, the different regions, the crown, the neck, the root. But here we do discuss a little bit more information. Understand there are 2 sets of teeth, right? So the first set of teeth that you get, right? First set, primary. The primary set are also called the deciduous teeth. Like a deciduous tree, we're kind of coming out of fall and now we're entering into spring, but there are trees that look kind of naked around town. Those ones are deciduous trees. They lose their leaves. And these are teeth that you lose as well. These are your baby teeth, also called the primary teeth. There are only 20 of these deciduous teeth, as opposed to 32 adult or permanent or secondary teeth that are going to emerge later and displace those primary teeth. So the primary teeth are going to come in first. They start coming in. Everybody's a little bit different. They start coming in about 6 months or so, but it certainly can come earlier. And what you start with, remember we have different types of teeth. We have our incisors, more medially, then our canines, then our premolar, and then our molars as we work our way towards the back. With the primary teeth, we don't have all representative types of teeth. We'll look at that coming up, right? Because we only have 20 instead of 32. But we start with our medial incisors, right? And usually you get your lower ones first, so you're going to see one tooth showing up there. Then you get a second one. Then there's a upper incisor that comes up, medial incisor, and then a second one, and then it kind of continues to branch out from there, right? The same order in which you get those teeth is the same order that you lose those teeth, right? So you start losing those. When you lose teeth, primary teeth, you lose them from the front, right? You lose one of those bottom ones. That's your loose one first. That one comes out. At some point, you may have all four of those medial incisors that are gone, a big gap there, so you can drink with a straw without even opening your mouth at some point. But those are going to be replaced and pushed out by our permanent teeth, right? They cause the root of those deciduous teeth to kind of degrade, go through apoptosis and things like that, and then they fall out. So when we look at it, right, so we can see here our deciduous primary dentition and secondary dentition. So here we see a central incisor, a lateral incisor, so we have two of those. We have a canine, and then we just have a first molar and a second molar. Whoa. All right, adult teeth, central and lateral incisor. You can call it medial lateral as well. There's one canine. Remember, it's 2I, 1P. And then we have, for the adult teeth, we have 2P, two premolars. No premolars in the baby teeth. And then we have 3 molars, 3M. First, second, and third molar, third one being the wisdom tooth. And those also, like we said, kind of come in at different time periods, basically earlier towards the front and then later towards the back. In the formula that we were talking about, the dental formula, that was referring to how many of each tooth type we have. 2, 1, 2, 3, right? So that adds up to 8. 8 over there, 8 over here, 8 over here, 8 over here. 4 times 8 is 32, right? In terms of dentistry and dental hygiene, of course, we need to keep track of the different surfaces of the tooth where a carie or some sort of a cavity might form or something like that, or a crack or crazing or something. And we need to keep track of what tooth has that so that we can watch it through time. So there's a specific numbering and lettering system to the teeth. So for baby teeth, we use letters instead. So you can see we've got in the upper A through J and in the lower K through T, like that. So you're seeing the upper right and then working your way around. And then for the permanent teeth, we use numbers instead. 1 through 16 on the upper teeth, up with the maxilla, and then 17 matches up with 16, and then curves around to go to 32. So the reason we use letters and numbers is, as we were talking about, at some point in your life, when you're younger, you're actually going to have a mix of adult teeth and baby teeth. And so it doesn't matter as much if you get a cavity on a baby tooth because it's going to be falling out, right? It does matter if you have some sort of degradation of an adult tooth. So they want to keep track of that. And so that way they can keep track of which are baby teeth, which are primary teeth, and which are secondary by using letters and numbers. Okay. So this we had already talked about, the different types of teeth. And of course, it's a good example of structure and function, anatomy and physiology, because we have different shaped teeth for different functions. The incisors and the canines are there for kind of biting pieces off of things. And if you're a predator, you have even longer canines because oftentimes you are grabbing a prey item and needing to hold on to it while it's moving around and stuff like that until it dies, and then you can eat it more. So you need these big tearing teeth that can hold on to your prey. Now our feeding style is not as that, and so our canines are not quite as long like that, unless you're a vampire. Then same thing. But then those, they have a hollow channel going through them. They go down to the root canal. It's a separate little, no, that doesn't exist. Anyway, I'm sure somebody went through the anatomy of vampires though, because right, they have to digest blood, a liquid diet. So how would that be in terms of their digestive system? Do they need a big stomach? Maybe, big volume. What about the small intestine? Maybe not as long, right? Because you're not starting with solid food. It's already liquid food. So maybe a short or small intestine instead. Who knows? Digestive enzymes? Maybe low-carb diet, right? High-protein diet instead. So maybe different enzymes too. Somebody's got to write that up. There was a cool book actually not too long ago. I have it at home. Somebody came up with, it was like for DC Comics, and they came up with like the anatomy and physiology of superheroes and stuff like that. So they have like actual diagrams, like a scientist who's writing up this big lab report of, okay, what's Superman's anatomy and physiology like that gives him those ability versus Batman versus, Venom or something like that, right? Venom takes in the solution, and I don't know what happened there. I probably pressed a button on this maybe. That changes his metabolism, what allows him to grow that way, and what more vascularization and capillaries and things. So it's kind of a good application thinking of things that we're learning and applying in different environments. Anyway, what? Oh yeah, sorry. Oh, it's okay. So different types of teeth for different functions. As we get towards the back, of course, we have broad malariform teeth. The broad surface allows us to kind of mash that in what's called mastication, really chewing it and smashing it in that physical type of digestion. And in fact, just another example of that, there are certain types, right? There are cartilaginous fishes. Do you guys know what cartilaginous fishes are? What are those? Examples of cartilaginous fish. So they have a skeleton, but it's made of cartilage. It's all cartilage instead of bone. Oh. So there's bony fish, true bony fish, and then there's cartilaginous fish, like the sharks and rays. So sharks and rays are cartilaginous fish. But when you look at the fish in terms of their dentition, and cartilaginous fish, if you're talking about sharks, look at their teeth. They don't have any premolars or molars, right? They're basically all big sharp things because it's all about grab and tear and then swallow whole. Not a lot of processing, not a lot of physical smashing in the mouth itself. Just swallow it and let the digestive system handle it instead, right? But there are things like rays that, as they're on the bottom, what they prey upon are like hard-shelled invertebrates, like clams and sea urchins and things like that, and snails that have a hard shell that they need to smash. And so if you were to look at their teeth, they have the opposite, almost all broad, flat malariform teeth for crushing that hard shell prey, and none of those fang-like or incisor-type teeth for biting off pieces of things, because they basically just like snap them off, right? So, in any case, it gives us an example, further examples of that structure and function for the teeth. All right, so we went through dental formula already. We went through the structure of the teeth already. So that's mainly kind of lab stuff, but we do need to know their structure. All right, so then we go from the mouth to the back of the throat, right? So the pharynx is the muscular back of the throat, and this is dual function, right? So it's both air passageway, as well as food and drink. So in this case, this is close enough to the outside. It's going to be lined with stratified squamous epithelium because we need to protect it. It's going to have mucus glands, mucus secreting, goblet cells and things like that in there as well, and has muscle layers to it, but skeletal muscle instead, because we said that it's going to be voluntary. It's when you decide that you're ready to finish processing something and send it on its way that it ends up going back. Now, interestingly enough, it's a little bit reversed in terms of the layers there. Don't worry, I'm not going to ask on the lecture exam, but it has an inner longitudinal and an outer circular. That's opposite of what we see when we get to smooth muscle further down, right? Everything we were seeing as we're reviewing microscope slides shows us inner circular next to the submucosa and outer longitudinal. So not only does the muscle tissue change from skeletal to smooth, but the orientation changes as well. So pharynx, and remember we have nasopharynx, but you shouldn't be eating anything through the nose, generally. Oropharynx is where we're going to start, then laryngopharynx. Larynx, with the epiglottis, is going to route food and drink posterior to the esophagus. So now we have this muscular tube, which is the esophagus, taking it down to the stomach. And esophagus also is a little bit different. Still has mucosa, submucosa, muscularis, but we have an adventitia. Remember that the serosa layer that we are constantly identifying is a serous membrane. And a serous membrane is when we get down to the abdominal pelvic cavity. Now they have a visceral peritoneum on the outside of those organs. That's the serous membrane. So this isn't down there yet, right? This is going through the chest cavity. So we have an adventitia on the outside as well. An adventitia is also what we have as an outer layer on the trachea. Both of those are right next to one another in that chest cavity. Okay? All right, so regardless, we've got the same layered look to it, but in this case, again, we have skeletal muscle at the beginning of it. So we have a muscularis, bless you, that now has changed over to circular and longitudinal like we saw before. But in this case, we've got our skeletal muscle transitioning into smooth muscle. So what we find is in the beginning here, with these organs, it's about processing in the mouth. But then when we get to the back of the throat, not digestion. There's not digestion going on there. There's propulsion, yep. There's secretion, yep, right? Mucus and things like that. But we're not doing a lot of digestion as we get to the back of the throat. In the mouth, ingestion, yep. Mechanical digestion, yep. Mastication is your fancy term that sounds naughty for chewing. You can masticate in front of people at the table. It's okay. So we have mechanical digestion. We've got chemical digestion as well. Salivary amylase and lingual lipase, a little bit of that fat digesting enzyme coming from the tongue. And then propulsion as well. So pharynx and esophagus is all about propulsion. You could, I don't have it written on there, but to be sure, secretion is occurring here as well, right? We have mucus being secreted all along the way. Okay, so after processing things, we decide to send it to the back of the throat. That bolus, you need to know what a bolus is. That's one of your vocabulary terms. We're going to do deglutition. Deglutition is swallowing. So we push that to the back of the throat. We can see how the uvula gets out of the way. Helps to protect the nasopharynx so that when you swallow, even if you were hanging upside down in trapeze or something like that, these things should go down towards the stomach as opposed to going up into your nose, right? Now, it doesn't always work well. So, you know, when we throw up, because y'all have thrown up as well, generally when we throw up, we have muscular contractions of the stomach, of the esophagus and things like that, that are sending it out here. Maybe it's a response to bacteria or something like that. Generally, your uvula is supposed to protect that as well, but sometimes it doesn't. Sometimes you get to enjoy it twice as nice, and it comes out the mouth and it comes out the nose. Yeah. So I don't know what the deal is there. Probably the uvula, in terms of its shape, doesn't fully seal off that nasopharynx. I know for me, unfortunately, it doesn't. I got a very narrow one. I don't know what the deal is. I'm not saying it's narrow. I'm just saying I think that's a problem. But I do know that if I throw up, I get some of it in there. I don't like that. It's bad enough you got to taste it and then you got to smell it. Yeah.
So is that true that that's one of the contributors to snoring too?
The uvula, in terms of uvula shape and things like that? To be sure, that's a possibility. A lot of that also has to do with the rest of that passageway, right? So any obstruction along that passageway is going to affect the movement of that air volume. So just like, unfortunately, we think about wind instruments, right? You have air going through a tube, and the diameter of the tube and any restrictions of that will produce a sound, and then change that sound. And so we're talking about restrictions. So it could be a nasal septum that's deviated, so the air is not going through properly that way. And then also as we get back to the pharynx and then down to the trachea, we've seen in the cases of our dissections and the cadaver that a lot of times if there's other tissues like increased adipose tissue there, it can impinge on these pathways and impinge on these organs. It's just layered right next to it. And so it can create a narrowing of it. And then the physical orientation of you, if you were sleeping straight up or something like that, it might be like standing up, then it might be better in terms of keeping that pathway open. But a lot of times when we're then laying down, some of these soft structures can fall back and kind of obstruct that as well. Yeah. Okay, so we're swallowing, it's going down, the epiglottis has covered our larynx, and we're sending it down to the esophagus itself. Muscular constrictions, peristaltic constrictions are sending it down to the stomach, and it's going to enter in this cardiac region, and there's also a little band there. This is a lower esophageal sphincter, also called the gastroesophageal sphincter that is there. So that's going to remain, for the most part, constricted, so that we don't have stomach acid going up into the esophagus. But if you're swallowing, that has to relax temporarily to allow this bolus of food to arrive. Okay? So then we get down into the stomach. So now with the stomach, there is a lot of both anatomy and physiology here. So the anatomy, I'm not going to go through it in detail, but we know that there's different regions to the stomach, right? We know that there's the rugae down the stomach that give it the wrinkles, give it the capacity to expand. We said potentially up to 4 liters. We also know that this is there for storage and physical digestion. So a lot of movement going on in there. So not only do we have circular muscle and longitudinal muscle, but there's a third layer, which is oblique muscle, that the fibers are going at an angle. So we don't have that in other layers of the digestive tract. So some unique adaptations that we see there as well: greater curvature, lesser curvature, etc. Pyloric sphincter regulating the movement down into the next section, which would be the duodenum. Okay, so what other unique aspects do we have to the stomach? Well, the epithelial lining of the stomach is somewhat unique. Because we have acid there, we all know there's hydrochloric acid down there, we need to protect it as well. So the epithelial lining of the stomach replaces itself very regularly, more often than other sections. It also, the mucus that is secreted there is bicarbonate rich. And so it's an alkaline mucus that is, at least on the surface of it, protecting the underlying epithelium, right? So it sticks to it very closely so that the hydrochloric acid and the gastric juice that comes out can mix with the food, but it tends not to then rest on the epithelium and digest it itself. We'll talk about what's coming out of those gastric glands, like we were talking about what's in gastric juice. Mucus, of course, is there, and then gastrin is, in fact, one of our first hormones that we're coming into that helps to control or regulate the activity of these organs. So anything that says gastro, etc., refers to stomach. Okay, so what we're looking at here is a very zoomed up version of the superficial layer of the mucosa, right? The mucosa right next to the lumen where the food goes. And we're looking at those gastric pits, like we saw on the microscope, that go down a little bit, not as deep as if we were talking about villi and intestinal glands. And then we have, instead of intestinal glands, we have gastric glands. So those cells at the bottom of those gastric pits make up the gastric gland. And there's a few different types of cells there. We've seen 2 of those in the microscope. Parietal cells that put out hydrochloric acid, and chief cells that put out pepsinogen. And pepsinogen is an enzyme, an inactivated enzyme, that then becomes activated to become the form that's called pepsin. Yeah?
What is it, or what's going on when you're bloated?
So, I mean, in that case, there's probably a few things going on there. When we are ingesting like a big meal or something like that, there's various things that are going on. Number one, you took in a large volume of something, so you tend to feel full. We have mechanoreceptors, kind of the feedback mechanism, that comes into play. The other things that we take in, the digestive process, depends on kind of what it is. We know there's different enzymes that digest different food molecules, etc. And there's also things that can potentially upset this system, right? So if you're taking in, as we talked about taste receptors, if you're taking in like very sour things or hot things or something like that, right? If you're doing buffalo wings or something like that, it has a lot of vinegar, has a lot of the capsaicin in it, the juices, extractions from peppers. So those things can irritate the system as well. And that can, or, you know, even other food products, right? You hear about beans and things like that, high fiber things that can produce gases that then can also produce extra stretching. So you have the volume of the food you took in, and then you have like stretching that can occur as maybe some of these secretions and gases start to mix together. In addition to that, and that can occur anywhere along the way, stomach, small intestine, large intestine, right? But then lastly, when you're taking these things in, they're being absorbed across, as we'll talk about coming up. We're bringing in nutrients, we're also bringing in electrolytes, we're bringing in water. And so as we bring those things in, that can also influence our balance in terms of our fluid concentrations, right? So the blood that we talked about before, we're loading that with nutrients, we're loading it with electrolytes. The water follows the electrolytes due to osmotic balance. So if you're eating a lot of salty foods, because sometimes people feel bloated after eating a lot of salty things, salty chips, crackers, things like that. As all those salts are absorbed, like they're supposed to be, right, digestive system is doing its job. Whatever you put in there, if it's small enough, we want it, we're going to bring it across. When you bring more salt across, the water follows. And so now our tissues are starting to gain more fluid as well. And so as our tissues gain fluid, that goes into the blood, and that increases blood pressure, but our tissues kind of get swollen. And so that also adds to a little bit of bloat, if you want, right? Sometimes it's water weight or whatever, and that often has to do with increased water ingestion, but electrolytes can influence that too, because all the solutes go across, and now we have an osmotic difference, osmotic pressure, and the water follows it. So a few different things can contribute to that. But all that's a really good example of what our system is doing, right? It's working to break things down small enough and bring them across the body wall, load it into the tissues, load it into the blood, and distribute it. It just takes a little bit of time for that to happen, and different things will be kind of loaded or absorbed differently. Okay, so different cells that we have down there. Parietal cells and chief cells are some of our most important ones, but there are mucus cells there too, as we were talking about. An alkaline mucus with bicarbonate in it to help layer this as well. And if we zoom up closer into these gastric glands, there are also what are called enteroendocrine cells in there as well. So as you can tell from that name, we've got cells of the endocrine system. So in a way, the stomach kind of counts as part of our endocrine system, as an endocrine gland that secretes hormones. Okay? So these particular cells down there secrete different types of chemical messengers, including things like gastrin. That's going to be one of the biggest ones that we're going to talk about. Histamine. Oh, we've seen that one already, right? So histamine can lead to increased leakiness in capillaries and cause fluid movement and things like that, right? So there's that. Endorphins. Oh, people hear about that. Right? Get happy because you're eating things, etc. Those travel in the bloodstream back to the brain. These are feedback mechanisms that either control things or allow the brain an awareness, if you will, about what's going on in terms of the stomach. This one, so endorphin, serotonin, right? Those are communicating back to the brain. This is another important one here, cholecystokinin, CCK. Now that one mainly is released also from the small intestine, and we'll talk about that one coming up also. But all of these are releasing chemical messengers. Now the interesting thing about it is that overall they affect these digestive processes. It can speed it up, slow it down. But that gastrin one is really interesting because gastrin, right? I said gastro refers to stomach. Gastrin is a hormone that's released in the stomach, and it actually affects the stomach activity. So when gastrin is released, it's basically affecting the other cells, like our chief cells and our parietal cells and things like that, and it increases the activity of the secretions of those cells and increases the activity of the stomach itself. So gastrin is a hormone, it's a chemical messenger, but it's not going far, right? It's basically having an impact on the same organ that it's being released from. Now it does go into the bloodstream, so it does have other effects, like it can get back to the brain, and the brain's like, oh, the stomach is cranking up its activity, okay, it's pretty busy here. I'll need to let the other sections of the intestine know that stuff's going to be coming soon, right? And it also can contribute, some of these can contribute to letting the brain know, oh, well, if the stomach's really active, that means I ate a big load of things. That means I should tamp down on my hunger, right? My appetite. If that's really full, mechanoreceptors tell me I got a lot of food in there. Chemoreceptors tell me what kind of food. These chemicals are being released. That's going to help to regulate how much food is coming in. Yeah.
So do those cells sort of contribute, like if you hear people say like they have comfort foods or they eat their feelings, is that that's why, like they feel like it's okay because it's releasing those endorphins and the serotonin making them happy?
Some of it. So some of it can come directly from the organ itself. But there's also kind of the influence of the higher complex areas of the brain, right? So what makes you happy, those chemicals can come back, but also there's kind of the brain influence down to the stomach as well. Like we talked about autonomic, whether you're stressed or not, also influences the stomach. So you can be eating chocolate, but gosh, I'm so stressed, I'm not digesting it very well because everything's slowing down because of sympathetic stimulation. So there's kind of like a top-down effect and a bottom-up kind of feedback effect that kind of all come into play. But, yeah, so it has a chemical basis to some of these things. It's not just straight, and even when we talk about emotions, right? Emotions are coming down to chemical messengers as well. So, okay. So as we mentioned, there's a lot of things going on in the stomach lining itself. We want to make sure that it's protected, as we mentioned, so that we've got a lot of mucus there. We've got epithelial cells that are kind of protected because they're linked together very tightly. And we have that alkaline mucus as well as kind of the gastric glands that are protected by these measures. Because inside of them, we've got a lot of cells that are doing things, whether releasing chemical messengers or hydrochloric acid or that pepsin that we were talking about. So we're trying to make sure that the stomach is able to do its job in a particularly harsh environment where we have that hydrochloric acid. So if there is inflammation there, for whatever reason, that's called gastritis, right? We still have the prefix gastro, and then we have itis, that's swelling, right? Gingivitis, when we were talking about the teeth, the gums are the gingivae. So gingivitis, swelling of the gums, commonly through a bacterial infection or something kind of rammed into it that you had eaten that was hard or whatever. Gastritis is where there's irritation down there. When you talk about ulcers, so ulcers can occur in a variety of places, but they do occur commonly in the stomach wall. And so this is where that inner lining has become kind of breached. It's not protected as well. And so now some of those secretions are starting to have an effect on the stomach wall itself. A long time ago, they thought, like if you have an ulcer, it comes from stress or something like that, right? Your job is too stressful. And so you have these depictions of people eating a lot of antacids. Oh, get that right now? Antacids are alkaline that are neutralizing the hydrochloric acid there. Now, if you have too much hydrochloric acid, then yeah, you can overwhelm the system. So you're taking these antacids, Rolaids, whatever, to help balance that out, right? But that will affect digestion as well, right? Hydrochloric acid is supposed to protect you against bacteria. It's supposed to help start degrading molecules and things like that. But more recently, while there might be an effect of that still, more recently they discovered that most ulcers are due to the presence of a high number of a particular bacterial species. So Helicobacter or Helicobacter pylori is a bacterial species that has in some ways evolved to withstand that harsh environment of hydrochloric acid. So it can still live down there where other bacterial species would kind of degrade and die off. It can actually live down there. So if you have, in essence, an infection of H. pylori in there, then that's also going to irritate that stomach lining and potentially cause an ulcer. So, again, more research certainly has helped us to understand how this system works. So there are pathogenic bacteria, right? Disease-causing bacteria, but we'll talk about later on when we talk about the colon and the large intestine, beneficial bacteria as well that are very important. Okay, so when we get to the stomach here then, this is the first place where we've taken in this load of food and we're going to be parsing it out like a tablespoon at a time down into the small intestine if it has gotten time enough to process, right? We need to be able to squeeze it and mix it around. If it's a high protein meal, if you're straight eating beef jerky for your whole meal, that's a lot of protein. Pepsin chemically digests protein down into peptides. And so if there's a lot of protein, it's going to need to stay in the stomach longer. If it's straight up sugar, right? If you're doing a smoothie or something like that, not a lot of protein there, a lot of sugar in there. Natural sugars or whatever, still sugar. So it will get through there. Not only does it not need to be physically processed very much, but not a lot of protein. So it's going to be going through pretty quick, absorbed pretty quickly and things like that. So the stomach, of what's going on in the stomach, does kind of set the tone a little bit for the whole system because we're going to be working on digesting that and working on sending it through the rest of the system over time. So in fact, there are 3 different phases of kind of control that go on in terms of our digestion there. One's called the cephalic phase. Cephalic means head. Another one's called the gastric phase. Another one's called the intestinal phase. So these are part of the alimentary canal, and of course that's the brain that's working to control them. So whatever is going on in any of these sections needs to be coordinated with the system overall. So in terms of stimulatory events to the stomach, if you're thinking about food, if you are smelling food or whatever, then our parasympathetic stimulation via the vagus nerve goes down to the stomach and starts it churning, right? right? Swallowing air, you're salivating more. Oh, now there's more fluid in the mouth. You've got to swallow more, which gives you a little bit of air going down there. Now there's air in there, and it's kind of causing it to distend, and so it's making it growl and things like that. So that type of information is coming where? It's coming from the hypothalamus. That is our appetite center. That gets the feedback from the rest of the body as to what's going on, and that is the control center for the brain, the hypothalamus. And then the motor output comes out from the brainstem, medulla oblongata, right? So we saw that for breathing. We saw that for heart rate and things like that, and blood vessel constriction, etc. So medulla oblongata, brainstem, is controlling this as well. So we see those parasympathetic stimulation going out because this is stimulatory. Parasympathetic is going to come out, and it's going to increase the stomach activity, increase secretions and things like that, increase motility, etc. So one of the things that comes out as well is gastrin. And so gastrin, what we find is that that increases the activity of the stomach itself. So that's kind of its local type of stimulation. The intestinal phase for the stomach, if we're getting it to increase, is basically the stomach says, or the intestine says, hey, you know what? I'm kind of empty, right? The mechanoreceptors say, I'm kind of empty. I'm ready for something. It also may say that we've got some items there that are, just finishing processing. Other types of food items like fats or something like that that are finishing processing there in the intestine. And it basically says, okay, stomach, when you're ready, I'm ready for your next load, okay? Now, if you go the other side of the coin, there's also inhibitory events because we want to make sure that everything is occurring at the right speed. Just like an assembly line, if you have one person that's working really fast and it's shooting it on down and those other people can't keep up, the whole thing's going to break down, right? Everything has to be coordinated. So, same type of thing. In case of our inhibitory events, maybe we have sympathetic stimulation coming down, or a lack of parasympathetic, and that's going to decrease the stomach activity. In terms of the stomach itself, if it's too acidic, or we have that sympathetic stimulation coming down, like we said, that's going to slow down the activity. And in terms of the intestinal, that's what this one is, if you remember. If that intestine is very stretched, or it's got a lot of acidic chyme in it from the stomach already, and it's got a job to do, it's going to send reflexes via the enteric nervous system back to the stomach and says, hey, slow your roll. Stop all that churning, just let it sit in there for a while, and once I'm done with what I'm processing, I will let you know, okay? And so we see that there's a variety of intestinal hormones that come into play as well. There's that CCK, that cholecystokinin as well, and secretin. Those are hormones that are released by the intestine and have an inhibitory effect on the stomach. So if the intestine is really busy, it's going to be sending out those signals saying, hey, I got stuff going on in here. If you can give me some help with that, like from our pancreas and our gallbladder and our liver, that's going to be a good thing. But for the stomach, you need to slow it down, and I'll let you know when I'm ready for more. Okay? So those are showing some of our kind of both neural, extrinsic, and hormonal, as well as local intrinsic control of the digestive system there. Now this one that we show here, don't panic. Focus on the pretty colors. Look at it as a bouquet of chemistry and just feel happy that you're seeing those colors. The reason I'm showing you this is not that you're going to have to learn it or memorize it or anything like that. But what this shows is our stomach lining here. Here's the stomach lumen where the food goes. We know that hydrochloric acid is released, right, by our parietal cells that we see there. We can see that in the wall of the stomach, here's our gastric pits, right? We actually have receptors for our different chemicals that can have an influence on the stomach, right? So gastrin, that hormone released by the stomach, can increase the activity. This is acetylcholine. Remember that for muscular activity potentially, as well as some of these biochemical pathways, histamine, etc. So we see some of the receptors that help to control the stomach activity coming into play. But what I wanted to show you is this. If you look at those little bubbles right there, what does that remind you of? Where did we see that before? a little chemical conversion like that. We saw it in the cardiovascular system. That occurs inside the red blood cells. Carbonic anhydrase is where we're converting carbon dioxide into carbonic acid and bicarbonate ions, right? So that little chemical pathway that we saw before in the blood cells also is so important, occurring up with the lungs, etc., when we're back converting it. But that is occurring in the wall of the stomach as well. Totally different system, but we're using the same chemical pathway. So we're taking bicarbonate that we see here, right? And that's coming from the carbonic acid that we've created from combining our CO2 with our water. So carbonic anhydrase in the wall of the small intestine creates bicarbonate and hydrogen ions. Now usually we're focusing on this, we're like, oh yep, now we can float our bicarbonate around, and that does go back into our fluids, etc. But the main reason that we have this chemical reaction going on here is that this is how we create hydrochloric acid. This is how the stomach creates hydrochloric acid. There's our hydrogen. There's some of our chloride as well that's being pumped across. There's our HCl. And HCl very quickly falls apart. The more hydrogen ions we're releasing here, the more acidic that stomach lumen is going to be. So in this case, we're using the same chemical reaction, but we're actually intending, if you will, to use that hydrogen ion. So it's not just like, oh, darn it, we made it more acidic, we got to deal with that. Here, yes, the intent is we made it more acidic, and in fact, we have to deal with this extra bicarbonate ion, which is called the alkaline tide. As we're secreting hydrochloric acid, we're retaining that bicarbonate, and now it's actually making our fluids internally a little bit more basic, a little bit more alkaline. We kind of got to deal with that as well. Okay? So that was just the point of this, is that's a mechanism. It's an interesting mechanism that we've seen before to produce the hydrochloric acid that we know that the stomach does. Okay. So, stomach is going to be doing its activity, putting a lot of secretions out, being very active, right? So, after about 1 liter of fluid or food that goes down there, that's when we start having those stretch receptors go off and the chemoreceptors going off and things like that. That affects its activity. And we have this ability to be able to stretch out and then have this elastic recoil. So the stomach is very plastic in terms of its shape. As it's digesting, it's going to be squeezing the food around like a washing machine, right? The churning that we see those 3 layers of muscle doing, they're going to be creating this rhythm of contractions. And in fact, there are pacemaker cells in the wall of the stomach that are in some ways analogous to the pacemaker cells that we find in the heart. So that's a natural rhythm of churning this stuff and mixing it around in there. Now, just like the heart, that rate can go up or go down based on sympathetic and parasympathetic in neural stimulation. But what we find is that it kind of looks like this, right? So here's a bunch, if you are vertical, a lot of your food is going to end up down there in the body and the pylorus. Remember, I have those different regions? Generally not going to be up in the fundus. And in fact, when they're doing x-rays, oftentimes they'll use the fundus to help them locate the stomach because it has air in it in many cases. And so on an x-ray, it comes in very black compared to our other organs that are more solid and have things within them. So they can see that fundus a little bit more. Maybe coming up, Thanksgiving and Easter, it might be totally full. But generally it's kind of empty there. So the food's down there, and we have these rhythmic contractions that are sending it down towards the pylorus. And kind of what it does is if that pyloric sphincter is closed, then what's going to happen is this stuff gets squeezed down there, and then it kind of splashes back. And it gets squeezed down, and it splashes back. So it has this washing kind of cycle, or almost like waves hitting the shore and splashing back that helps to mix our different secretions with the food items that are there. Now, at some point, that pyloric valve is going to open as well, and we're going to let just a little bit of that acidic soup called chyme. That's one of your vocabulary terms. The acidic soup of the food plus the gastric secretions is called chyme. It's going to let a little bit of that out so that it can go down to the small intestine and be further digested. Okay? So, a lot of different things coordinating that activity in the stomach. From the brain, cephalic, from the intestine, from down below, as well as from the stomach itself. So we're seeing the effect of hormones, different types of chemicals that are released, like from the duodenum. And then, again, different types of neural control, enteric nervous system, as well as extrinsic neural control as well, right? Parasympathetic increases, decreases, and things like that. Okay, so stomach was a big one. It's a big organ. There's a lot of different cells there. They're secreting things. If we focus on what was going on there, definitely secretion, definitely propulsion, definitely chemical digestion. Chemical digestion of what? Is it carbohydrates? Is it lipids? Is it proteins? Proteins mainly, right? Pepsin is released in the stomach, and it works to break peptide bonds. It's kind of in the name, right? Break them down, proteins down to peptides and amino acids. So chemical digestion going on there in the stomach too. When you're a baby, interestingly enough, you have gastric lipase being secreted as well. What does lipase digest? Lipids, fats. What are you ingesting as a baby? A lot of fat-rich milk, right? So there's a lot more milk in your diet, a lot more fat in your diet that needs to be digested. So we actually have more enzymes that helps us to break down that, and then it kind of decreases through time, right? All right, so let's get down into the small intestine. Before we do, questions? Stomach.
Can you review again I was trying to write the modes of what's
Control?
secretion,
Oh, okay.
chemical digestion of proteins, propulsion.
Propulsion's there.
Is
Mechanical digestion, we
that's
can
what I
say
was
as well. The churning that goes on in the stomach, that splashing and stuff like that, that churning also counts as mechanical digestion.
Okay.
Physically kind of squishing things around as well.
I wanted to make sure.
Yeah. Okay. So small intestines, 3 different subdivisions. First one is what? Starts with a D? Duodenum, then jejunum, then ileum. Okay? Now in the microscope and in the models and stuff like that, we can easily tell our duodenum and our ileum because that's the beginning and the end. Jejunum's kind of in the middle, and so that one's harder for us to really identify, but it is the middle section. It's a very long section, but it's the middle section. So with this, we're going to talk about, for the most part, duodenum to begin with, and then jejunum and ileum, those are really about the finishing of the process, the actual digestion, chemical digestion, and absorption going on there. The duodenum is kind of a mixing area instead. So we're going to find at the duodenum, right, we have the common bile duct coming in, and we have the pancreatic duct coming in. We have these accessory organs that are releasing their secretions at the duodenum to mix with the food again. This is like we said, like the dishwasher releasing detergent to mix with the dishes which have the food on them. A lot of mixing that's going to go here at the duodenum. So there's our duodenum. It's our shortest section. This is like 10 to 12 inches. We said that the small intestine can be anywhere from like 12 to 15 feet long. And this is only 1 foot. So the duodenum is definitely the shortest section of it. But this is where we're going to have that mixing going on. Now, in terms of the structure, to a certain extent, our different sections of the small intestine will have those adaptations that we've been learning about, right? Plica circularis and villi and things like that. Even those will change a little bit as we continue on. The villi get a little bit longer, more surface area, etc. But this is our duodenum. We can see some of those circular folds occurring in through here, right? We can see our duodenal papilla, that little bump that we can see on a couple of our models that controls the release from the common bile duct and from the pancreatic duct. So anywhere in our small intestine, like we said, we'll have those adaptations that increase surface area. Plica circularis, also called circular folds, villi, as well as microvilli. So we didn't see the microvilli in the microscope. We probably got to about this level, right? So there's a plica circularis. There's one there, right? Each of these are the circular folds. And then these little finger-like structures extending out are the villi on top of that. But then if we look at an individual villus, right? Villus is going to be, so villi is plural, so this should really go like this. If that was truly marking more than one, villi is plural, villus is singular. If we're pointing to one, we would say villus. So on these simple columnar epithelial cells here, line the edge, just like our intestinal wall model, like we see in the lab. These simple columnar epithelial cells actually have microvilli on them, again and again and again, adding even more surface area, right? So it's like those Russian dolls, those little dolls that the smaller one and smaller one and smaller one. Everywhere along the way, we see these adaptations for more and more surface area there so that we can digest and then absorb. We also see some of our structure in terms of duodenal glands showing up there, our lacteals, which are aiding in the absorption of fats. So remember that as we're digesting fats, that process is going to be a little bit more complicated, and sometimes those things that we're absorbing are a little bit bigger and easier to get into our lymphatic capillaries or our lacteals there. Okay, so we see a variety of things in there. Our villi, our mucosa, submucosa, muscularis, and serosa. Only 2 layers to our muscularis, inner circular and outer longitudinal there. We do see kind of duodenal glands, Brunner's glands or duodenal glands. Duodenal is a little bit easier. It makes more sense to say duodenal as opposed to Brunner's. And we also find our lymphatic or Peyer's patches there as well. Those are more common to find in the distal ends of the small intestine where we're actually doing absorption. Because at the beginning of the small intestine, like the stomach, we haven't had a lot of chance to do digestion yet. We haven't gotten things down small enough to absorb them across the body wall. So for instance, even in the stomach, where you're like, oh, my stomach's full and now it's empty, I guess I just absorbed all that stuff, right? Nope, it shunted into the small intestine. So there's very few things that get absorbed in the stomach. You can absorb things like some water and electrolytes. Alcohol can start to be absorbed across the stomach wall and stuff like that, things that are already kind of small. But for the most part, there hasn't been enough time and space for things to be broken down small enough yet. That's going to occur in the small intestine instead. And so that's where we need our protection, because remember, Peyer's patches or lymphatic patches or nodules are part of our second line or even third line of defense, right? So we're going to have different types of phagocytes in there, lymphocytes are in there, in case things come across the body wall and try to infect us. So we tend to find those in the last sections, like in the jejunum and the ileum, more so. All right. So we said that the duodenum, that first 10 to 12 inches that we see foiling right through there, is going to be a mixing point for the secretions from the gallbladder, from the liver, and from the pancreas. So we'll kind of address those organs while we're here. So we're going to start with liver and gallbladder. Liver is our largest kind of cohesive internal organ. So it's about 3 pounds or so. Has 4 different lobes to it. You don't have to know the different lobes, but it does have 4 different lobes divided by that falciform ligament that you can see how it's connected to the diaphragm, right? It still has to be connected and suspended. The main function in terms of digestive function is that this is the organ that creates bile. The liver makes the bile, and the gallbladder stores the bile, right? Stores the bile instead. And bile is what? What was that E term that we said bile is? It's an yeah. So it's an emulsifier, right? So it does emulsification. It helps to physically break down large fat droplets and separate them into smaller ones, okay? So that's its main role. It's created by the liver, and then it is stored in the gallbladder. So if we're digesting very fatty foods, we're going to need the help of that bile from the liver and the gallbladder to do so. So we saw in the microscope that the cells of the liver are called hepatocytes, and they create these little liver lobules. So each one of these units that we see here is going to be a liver lobule, and all of those different cells there are hepatocytes helping to make it up. So in the center, we've got a central vein. So we can see how this is going to be gathering up blood and then routing it towards the inferior vena cava ultimately. Along the periphery, you can see that there are these little triads as well. So there are hepatic arteries. There are branches, in fact, of our portal veins. There's little portovenules. And then there are little green vessels here, which are bile ducts. So the hepatocytes make the bile, and they get secreted into bile ducts that are at the periphery. So you can see the bile goes this way. The blood, as it's being processed, right, because this nutrient-laden blood that's been absorbed in the intestine, right? We've done the digestion down further in the intestine, and now we have all of these nutrients in the blood. A lot of those nutrients we may want to store, or we may want to process further a little bit. So like our glucose that goes into the bloodstream, if we're not using it, a lot of that gets stored in the liver cells, in the hepatocytes as glycogen. And so the hepatic portal-vein is taking this nutrient-laden blood from the small intestine, taking to the liver so the liver can continue to process those nutrients and potentially store those nutrients as well. So that's why we see those portal veins there in addition to the regular veins, okay? So lots of different things that our hepatocytes are going to do in addition to production of bile, processing bloodborne nutrients, storage of fat-soluble vitamins, detoxification, which is good for you. That's why they talk about, right, if you have a heavy drinking binge, right, that's hard on your liver because that alcohol gets absorbed and we need to detoxify it, etc. So there's like 30 different functions for those hepatocytes, but those are just a few of the major ones. And in terms of digestive processes, production of bile to help break down fats is the main job of those hepatocytes, okay? So it's kind of simplifying it, but we're focusing on, okay, how is the liver contributing to these breakdown steps for food itself? Okay, so the bile. The bile is this kind of greenish yellow fluid that contains a few different items. It contains bile salts. As we saw in the PhysioX simulation, those are the salts that do the emulsification. On the right-hand side is a little figure there of how those salts will attach to the fat droplets. They help to break up the fat droplets, and then they attach to it and keep them from reforming again, right? Because if you've ever, like we talked about, had fat like droplets on the surface of the water of the sink, if you were to like mix your hand around it, or if you had like salad dressing, right? It's like an oil and vinegar salad dressing. If you see that stored, you can see how the oil and the water separate out. And so what do? You're like, oh, I don't want just straight oil, so you mix it up. So physically, you can break up those larger fat droplets into smaller ones, and now everything kind of flows out together. But if you let it go back together, hydrophobic, hydrophilic, it will sort out again. And so those bile salts adhere to the fat droplets and keep them from coming back together. So they emulsify the fat. There's that term that we were talking about. And in that way, they kind of facilitate the fat digestion as well as absorption. Interestingly, cholesterol comes into play too. So cholesterol is a component of bile as well. And so that's one of the ways if you get, like you hear about gallbladder is going to be storing the bile. If we get a solidification, if like there's a lot of cholesterol in there or something like that, too much, you may get it going from liquid form to a solid form, and now it can't move very well. So when you talk about gallstones, etc., that can have to do with an imbalance in terms of the amount of cholesterol and the fluidity of that bile that we have there. The chief reason that the bile looks the way it does is that has to do with bilirubin, which remember that one of the other roles of the liver is to take out worn out red blood cells. And we do recycling of the hemoglobin and stuff like that. Well, when we're processing that heme group from the hemoglobin, some of that pigment that we see there gets converted and it takes on this greenish hue. So bilirubin is a byproduct of processing that hemoglobin, and then we get rid of it by releasing it into bile. Now, in fact, some of it gets reabsorbed along the way. We get it back again, but that's part of the process of what that bile is doing as well. So liver makes the bile, gallbladder stores the bile. So super easy in terms of a function for the gallbladder, right? So whereas liver is doing secretion and even some propulsion, if you want to consider that, because we're squeezing the bile out, gallbladder is all about secretion or release of fluids that it didn't create and then squeezing them down to release into the duodenum. So we have the cystic duct, which drains the gallbladder itself, so we see here. And that combines with the hepatic duct coming from the liver to form our common bile duct that drops down into the duodenum itself. So it stores and concentrates the bile itself. It absorbs some of the water from it, right? Because it's easier to transport it from the liver to the gallbladder when it has more water from it. And then ends up concentrating it so that what is finally released down to the duodenum is that kind of thicker bile instead that's kind of mixed with our food. All right, so we had liver, we had gallbladder, now we have pancreas. So pancreas, feather-shaped organ that's deep to the stomach. Pancreas is super important. Pancreas is also interesting because it is an exocrine gland, right? Produces a secretion that gets released into the local environment through a duct, and it's an endocrine gland, right? Everybody knows that the pancreas is where we make insulin and glucagon and a couple other hormones, right? But that counts as an endocrine organ instead. And as we saw on the microscope, remember that we have the pancreatic acini. Those are the exocrine function ones that are producing the pancreatic juice. And then we have the pancreatic islets, which are the little islands of cells that are endocrine instead. So what it's going to be doing is it's going to be producing its secretions and releasing them into the duodenum. So if we harken back to what we saw, we have acidic chyme coming down from the stomach. It's going into the duodenum. Now that acidic soup would eat right through the duodenal wall unless we neutralize it. So the secretion from the pancreas has a lot of sodium bicarbonate in it, which is very alkaline, and that helps to neutralize that hydrochloric acid. In addition, within the walls of the duodenum, remember we have our duodenal glands that are only found in the duodenum. That's why they have that name. And they're producing an alkaline mucus to release and then mix with that acidic chyme to help neutralize it as well. So we're neutralizing that acidic chyme that's coming down. We're making the environment more of like a pH 7, and the pancreas helps with that. The duodenum does it itself with duodenal glands, but the pancreas helps with that as well. Okay, so as that food enters into the duodenum needs to let these accessory organs know, hey guys, I got this new stuff, this new food I got to process. I'm going to need your help. So I'm going to need the gallbladder. Give me some of that bile. Liver, keep making that bile and give it to me. And pancreas, I'm going to need your pancreatic solution or pancreatic juice there. So sodium bicarbonate to neutralize that acidic acid. But then also pancreatic enzymes that we're going to be talking about. More enzymes coming in.
So the pancreas releases secretions that reduce the acidity of what? What is it called?
Chyme. So CHYME that we have there in the upper left-hand corner. So that is the term for the acidic soup, if you will, that's coming from the stomach itself. So when the duodenum gets that acidic soup, one of the hormones that it releases to communicate that out is secretin. So its name is pretty straightforward, right? So secretin, released by the duodenum, tells the pancreas, hey, can you secrete your pancreatic juice to me to help neutralize this acidic chyme that's coming down? So when the pancreas receives that secretin, then it starts putting out more of that pancreatic juice to protect the duodenum. So it's a nice little chemical communication pathway that doesn't have to go too far, but it has an effect on it. And in fact, that same secretin, when it's released, can also have an inhibitory effect back at the stomach as well. Okay, so this is just the histology of the pancreas like we saw before, right? With our pancreatic acini, these are exocrine cells that are putting out the enzymes in the pancreatic juice. And then our pancreatic islets, these are the ones that are putting out our hormones that help to regulate blood sugar instead. So what we're looking at for the pancreas is it helps to neutralize the acidic chyme because it has that bicarbonate in it. And it's going to be putting out a bunch of different enzymes. So the enzymes that are released, what we've seen so far is that we'll see, oh, okay, there's one enzyme that comes from the tongue. There's one enzyme that comes from the salivary glands. There's one enzyme generally that comes from the stomach. So it's been kind of piecemeal to this point. We're going to do a little bit of carbohydrate digestion, a little bit of protein digestion, etc. Now it's like we need everything. We've reached the small intestine. We need to break down every single food item that we've ingested. So in fact, you've got proteolytic enzymes for proteins. You've got pancreatic amylase for those carbohydrates again. You've got pancreatic lipase for our fats again. And you even have nucleases for breaking down our DNA and our RNA. So we've got an enzyme for every single different type of food molecule that we've ingested, because this is where we need to chemically break them down again. So lots of enzymes coming in, secretin coming in, and CCK. So CCK, like we mentioned, comes from the stomach and from the duodenum, and CCK from the duodenum tells the pancreas to release those enzymes instead. Okay? So secretin controls the pancreatic juice, the bicarbonate, and the CCK, this cholecystokinin, tells the pancreas to release its enzymes, because they each have a function to do. So lots of secretions for the pancreas. So it's a really, really critical organ, an accessory organ that we have for the digestive system. Okay, so we'll finish up with pancreas next time, as we were talking about the roles of those hormones in controlling pancreatic secretion. And then from there, we've done liver, gallbladder, and pancreas. We're at the duodenum. We'll finish up with small intestine, and then we'll move to large intestine as we are getting through our alimentary canal. So as we finish up that, then we'll do a review of kind of that chemical digestion of the different food molecules and finish up the digestive system next time. Okay?